Chapter 8, Section 2, Chromosome Replication and Segregation. Now, for any and all living cells, before they can divide, there has to be replication or copying of the chromosomes. Okay, makes sense. All resulting daughter cells have to have a copy of the genome. So, what have we done so far? Us being scientists. Well, we've studied this in E. coli. Okay, E. coli, readily available, easy to culture, easy to work with. What we have found so far is that there is a spot, singular spot on their chromosomes that replication of the DNA chromosome begins. They call this the origin of replication, or the ORI-C, O-R-I-C, ORI-C. What happens here is a specific protein binds there. DNA A. DNA is the gene, or the gene family, and then A is the specific one. Just like ORI-C, O-R-I, ORI, is the gene collection of genes that have to do with the origin, and C is the specific gene. Notice it's the 3 letters and then a capital letter. Okay, when you see like this right here, there it is, ORI-C, lowercase, uppercase, this denotes you're talking a DNA or gene sequence. If it's capital, like here, capital DNA A, capital A, this is the protein product of the gene, lowercase DNA, uppercase A. All right, so keep that in mind. So when you're looking at stuff and you're not sure, am I looking at, and is this discussing a gene, or is this discussing a protein or an enzyme? Who's in uppercase, who's in lowercase? Supposed to be all genes, lowercase, uppercase letter, and italicized. If it's not italicized, uppercase, uppercase, gene, or it's gene product, also a protein. Could be an enzyme, could be a structural protein, whatever. So anyways, back here, ORI-C, notice lowercase, italicized, so this is a DNA region. You have the protein, DNA A will bind here and will kick off this. So you have the ORI-C. Sorry, I had to stop there. Dog wouldn't shut up. The ORI-C, this DNA region. Okay. You have a protein, DNA A, that's going to be binding to it. That's going to kick off. What it does is it helps start the insertion of the proteins that are going to form what is known as the replosome. Remember, that's going to be the DNA polymerase, the gyrases, excuse me, single-stranded binding proteins, all of that good fun stuff. DNA A is most active when it's linked to ATP. Not phosphorylated per se, but when it is literally linked to it. DNA A, ATP. To inactivate it, well, there's a couple of ways to do it. First, competition. If DNA A, ATP can't bind to ORI-C, no initiation of replication. Another way is to stop replication of DNA A. Okay, DNA A activity, not only is it going to be ATP, but it's going to be a transcription level event. DNA A levels is going to go up and down in the cell. High levels, or its presence, you're going to get DNA replication. Low levels, no replication. So, go after the expression. No transcription of the DNA A gene, no DNA A proteins will be made. Another thing is to, titrate it away. Have something else around that will bind up DNA A. So yeah, you have a certain amount of it, but if a lot of it's caught up over here, bound to something that shouldn't be, and then OREC's over here, it's all stuck here, none of it's getting here, no replisome. And then finally, well, if DNA is active, most active when there's ATP there, cut off the ATP. Done. 4 ways to regulate DNA activity on OREC. Turns out the double-stranded helix, that's the bacterial chromosome, is methylated. Okay? It's methylated. Means it has methyl groups added. I do believe it's on the phosphodiester backbone. Don't quote me on that. Well, if both strands are methylated, when the strands are opened up at the replication bubble, and then you get the new strands laid in, new strands haven't been methylated yet. Only one strand of the double helix is methylated, so you end up with what is referred to as Hemi Methylated DNA. Hemi methylated. Partial methylated. Doesn't mean partial and some here, some there. It means only one continuous strand of the double helix is methylated, the other is not. Turns out DNA A does not like to bind to the hemimethylated DNA. Protein known as we're just going to call that sequence A, CQA. Yeah, so that's what we'll call it, CQA protein. This has an affinity for this hemimethylated DNA. Also, turns out CQA also has a much higher affinity for hemimethylated OREC, which DNA ATP kind of, sort of, but not really has an affinity for. So this will block this, which makes sense because if you're in the process of making a new double helix, you don't want to start making more double helixes, so you're going to use this methylation event. Start off with one double helix that's fully methylated, both strands. In the process of replicating it, you get 2 double strands or 2 double helices that are somewhat methylated, hemimethylated. Only one of the anti-parallel strands will be methylated. Takes time after both those chromosomes are finished for the enzymes to go through and fully methylate it, making it fully methylated. It's kind of a timing thing. This is how the way so that the cell knows chromosome's either not done replicating yet, so don't start at more, and 2, we haven't divided yet. We're not ready for any of this yet because, well, not everything's been methylated. So when you look at it, a lot of times people who have a background in eukaryotic cell biology want to sit there and, relate things to, what they know. Oh, I know mitosis. So, all right, mitosis. G1, S, G2. All right, so the analogous processes that you'll find in a prokaryote. G1, cells being a cell. Okay. S phase, cell still being a cell, but it is all also replicating. G2 of the eukaryotic cell cycle, well, chromosomes replicated, get all the machinery ready to divide. That's what we see here. This thing, this circle here, they call it Z ring, and we'll talk about what the proteins involved there in that Z ring. It's a contractile ring that will draw in the cytoplasmic membrane, the peptidoglycan cell wall, bring it in, and pinch off. And then you get the cell division. Does everything fit up exactly like it would if this was a mitotic cell? No. But this is just a way so we can look at it and draw comparisons. So what's happening during the S phase, that synthesis phase? Well, under a cell that has had time to replenish, get all the nucleotides back up again because it divided 15, 30 minutes ago, whatever. The chromosome has been completely methylated, as you see here. The little red hemispheres are methyl groups. They go up and down both sides of the double helix, both strands. This has an affinity for, or DNA has an affinity for this kind of DNA. All right? Has an affinity for this kind of DNA. And specifically, right here at the ORIC, the origin. Nearby here, you're going to have the gene for DNA A. Notice this is lowercase, so this is the gene. Here's the protein product. So DNA A is going to interact here at the ORIC, and at the same time, it's going to upregulate its own expression. As you get the replication bubble forming, and remember, it's going to be expanding the replication forks at both ends. Well, this new DNA here, only one strand, the original template strand, will be methylated. The new strand being laid down, not methylated. So this is where they call Hemi Methylated. DNA A has little to no affinity for this form of DNA. Turns out sequence A, seq, I don't know what we're going to call this. We're just going to call this SEQ. SEQA. Okay? No funky names for it. SEQA has a high affinity for this, which means it's going to want to bind here. By binding here, it stops DNA A, so it stops the creation of a new replication bubble. And at the same time, it stops transcription and the making of more DNA A. So, the binding of SEQA represses DNA expression. Cool, all right. The protein, DNA, its expression is autoregulated by itself, binding promoter boxes. Promoter boxes upstream of the DNA gene. Okay, so not only does it start the formation of the replisome and the formation of the replication bubble, but it also regulates the expression of itself. Kind of cool. HDAA, which is an ATPase, will hydrolyze ATP or DNA ATP. By hydrolyzing it, what it's doing is it's cutting right there, releasing. So now you just have DNA A. DNA A, remember, while it does have an affinity for fully methylated DNA, it is greatly reduced. So none of this is completely an on and off, but it's degrees of just how on is on and just how off is off. As you learned in Intro Cell and Molecular, hopefully you saw it in Cell or Genetics, When it comes to the replication of DNA, it's not just in one direction. Okay, it's going to be bidirectional. It's going to start in one place and then it's going to go out in both directions. Okay, because of this, the replication can take about 40 minutes, start to finish. It's not too bad, but it's kind of weird seeing as it's a 20-minute generation time. Replication takes twice as long as the generation time. Well, it means that as the cell's dividing, it's already in the process of making new chromosome for the next division that's going to be coming along in 20 more minutes. So because of this, you can end up with multiple DNA forks. Remember, I told you DNA A has a low affinity for hemi methylated, but not a complete lack of affinity. So if it was just one single replication fork occurring, you start at one end, you go, it would take 3 times as long to go from 1 to 2 cells. And those cells, well, they're kind of stuck. But by having multiples occurring, remember you're dividing every 20 minutes. 20 minutes, 20 minutes. So you start with 1 and 40 minutes later you've got 4. But look what's happening. 1 replication fork, and then each of those resulting are going to act as template for more and more. So here, by doing it this way, our bottleneck, our limiting factor here, is how quickly the cells can replenish amino acids and lipids and phospholipids so that it can make more cell wall. Not how quickly can the chromosomes be replicated, because we're ready to divide now. However you're doing this, you got to remember, every resulting daughter cell has to have 1 complete chromosome. So our bacterial friends have spent the past billion years or so evolving to utilize what is known as the PAR system. PAR for partitioning system. It's a series of proteins and whatnot that will ensure both resulting daughter cells have the chromosome. So as the chromosome is being copied, it's being pulled apart and separated physically. PAR A, ATPase, PAR B, which is a chromosomal binding protein, POP-Z, PAR S, all of these will coalesce, form a complex around ORI-C. POP-Z will localize to an old pole, anchoring one of the chromosome there via PAR B bound to S. Because remember, the PAR S is going to be here, POP Z is going to be anchored. This will allow, so we have, here's the old, and as the new is being made, it can be pulled away. PAR S binds to another complex of PAR B, and PAR S is what's going to be pulling away the new chromosome. All right, let's go over here to the new section, or let's go over here somewhere else so that we can get some distance. Now, this whole PAR system that I just mentioned on the last slide, this is E. coli independent. All right, E. coli does something completely different. E. coli uses a topoisomerase 4, and then it uses the MUC-BEF system. MUC-BEF, sorry. MUC-B, MUC-E, MUC-F. This is a complex of proteins that work with topoisomerase to, as the new ORI-C is made, they'll just be pulled across to, terminal ends of E. coli so that E. coli can just partition down the middle. That whole system we talked about on the last slide, that's going to be for something like Colobacter here, where you're going to have the original, and then you're going to get a little budding extrusion that's going to become the new cell. Well, how do you make sure that happens? Well, you have your anchor point right here, which is pop Z. Pop Z via par B and par S is going to adhere to the chromosome. The little arrows you see right here and here, this is your replication forks. So as the new chromosome is being made, you have, here the terminus protein. It is being pulled right here, boom, boom, boom. Here's the new origin. There was the original origin. Par S bound to par B is acting as a motor protein and pulling this way. Remember, our two replication forks will meet here at near the terminus. So as they are moving this way, the new DNA is also being pulled, or the new double helix here. One of the new is being pulled here. The other is being held in place. All of this that we see from there up is going to become a new cell that floats away, will form its own stalk in the future, and then become sessile itself. E. coli is doing something completely different. E. coli, you see here, the yellow. Yellow is going to the origins. The Col E is the plasmid showing, hey, here's what if we do use a plasmid, because plasmid's going to be just like the chromosome. It's going to be copied, and then it's going to be transported to the terminal ends. Here's where the original nucleoid is, and then you see it's going to be pulled apart, and then at the near future, cell wall partition is going to cut it in half. Cut that there. Cut there. And so on.